The mission of the European Medical Information Framework, known as EMIF, is to improve identification, access, and assessment and reuse of health data within the European Union. With this mission in mind, access to descriptive information about the data available in EMIF was expanded to researchers beyond EMIF. Two tracks in the EMIF architecture support two data types: electronic health records, which include routine healthcare data from general practices and hospitals, and cohort datasets, which include data for specific research communities. Let's explore how you, as a researcher, can utilize cohort data. You may have a specific study in mind. You'd like to utilize the real-world data accessible via the EMIF platform. The EMIF process is oriented around disease communities. If a community for your disease state does not yet exist, you can establish a new community by leveraging all the tools of the platform to facilitate collaboration and data sharing. An important step during onboarding of new cohorts is the harmonization of data. EMIF has developed the technology and governance framework to support this process. If your desired community does exist, you can apply to join it and start browsing for interesting cohorts. You can then use the participant and variable selection tools to identify subsamples of individuals and subsets of variables that fit the needs of your research question. You can even filter based on variable value. Once your data access requests are approved by data owners, you will be able to conduct private, secure, remote analyses. Your first stop is the EMIF Cohort Selection tool. Select the community for the disease area relevant to your research. Let's dive deeper into the process using the Alzheimer's disease cohorts as an example. Use the Cohort Selection tool to review cohort information. You can browse individual questionnaires and use search features to compare cohorts and assess suitability for your research question. Then log into the Participant Selection tool and see how different cohorts can contribute to your study as you change the inclusion criteria. Use the Variable Selection tool to check which analysis variables are available for a particular group of patients. These initial steps allow you to establish the feasibility of your research question and enable you to develop a study protocol. You are then ready to submit a request to the relevant cohorts to access their anonymized patient-level data. Data owners approve access to their data based on your study protocol, requested patients, and the analysis variables. They each decide whether to approve your request. Once consent is granted, data from approved cohorts and analysis tools are provided securely in a private remote research environment, for example, using a data platform called Transmart. Researchers can use Transmart to access data, perform statistical analyses, and produce figures. For example, you might compare demographic characteristics and cognitive scores of APOE4 carriers to those of non-carriers. You can also log into the private remote research environment and perform analyses using your own tools. Once analyses are completed, you will prepare a manuscript for publication in a peer-reviewed journal where you must acknowledge all EMIF partners of the study and include data owners as co-authors. We encourage you to advance your research by becoming part of existing communities and helping to develop new communities within the EMIF platform.